Hello and welcome to BioRules video lessons. We are starting now the last class on biochemistry of nucleic acids, where you're going to see the process of translation. So translation is the process where we change the language. So in this case here, we are going to be changing from nucleic acid to another medium, which is protein. So basically, translation means getting nucleic acid information and using that to produce proteins. So that's everything that we have been doing so far. DNA to RNA, the transcription is to make RNA, because RNA then is going to be used to make protein. So basically now we are going to be getting this RNA, this messenger RNA, and we're going to be using that information to be making protein. So this here is the process that we are doing. So we had so far the transcription, where DNA becomes RNA, and now we are going to be getting the RNA, which in eukaryotes are going to be inside the nucleus. It leaves the nucleus, go to the ribosome in the cytoplasm, and is going to be making the protein. So let's take a look on how translation happens. The first thing for us to understand translation is to understand that it's going to happen by reading messenger RNA. So the messenger RNA contains information. A, U, G, U, A, all those letters, they are going to be coding for proteins. And the question is, how this genetic code, how these letters are informing the ribosomes, our cells, which is the amino acid to put in which place. So the first thing, for us to understand the code, the genetic code, is how many letters are going to be necessary for you to produce something that says this amino acid or that amino acid or that other amino acid. So let's start thinking about one-letter words. If you have one-letter words and there are 4 different type of letters in the messenger RNA, A, U, C, or G, then what we're going to be having, or A, or U, or C, or G. So in that case, there would be 4 possible combinations. In this case, 4 possible information codes. If you have A, you have one information, U, you have another information, C, you have another information, G, you have another information. So if the code was made by one-letter words, we would be able to code for 4 amino acids. That's not enough. We need more than that. So what about 2-letter words? So we're going to have there AUCG and another one AUCG. So in this case, that increases the number of possible combinations that you can have, because we're going to have 4 on the left, 4 on the right. So I can make A, U, A, C, A, G. U, A, U, 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 C, U, G. C, A, C, U, C, 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 G. G, A, G, U, G, C, G, G. So in this case, I can make 4 times 4 combinations, which makes 16 possible combinations of letters. And this is almost enough. Now 16 combinations, it's pretty close to what we need, but it's still not enough, because we have 20 amino acids to be coding. So 2 letters is not enough. So let's take a look now on the next possibility, which is words of 3 letters. If the words are of 3 letters, we can have 4 in the first one, 4 possible different in the second one, 4 possible different in the third one. So you can make, for example, AAA, AAU, AAC, AAG, AUA, and so on. So basically you can have 4 possibilities in the first, 4 possibilities in the second, 4 possibilities in the third. Is 4 times 4 times 4. This makes 64 different combinations. And 64 different combinations, that's more than enough for us to be able to code for all the amino acids and all the information that we need to make translation. So the code, the RNA code that comes from the DNA, but the RNA code that's going to be coding for amino acids is composed by 3 letters, which we are going to call it a codon. Okay, so we have here this kind of information showing us the DNA that you have transcription making here, RNA, GGU. This RNA here then is read, and it's going to be making, for example, a G, and a U in the messenger RNA is going to be coding for a glycine. while an A, a G, and an A, an AGA, is coding for an arginine, and an A, a C, and a C codes for a TR. If I'm not wrong, that's tryptophan. So different three letters are going to be coding for possible different amino acids. This is the idea of the genetic codes using these three letters words that we call it codons to code for amino acids. And here we have the star of the show. This is the table showing how we combine three different letters making different amino acids. For example, the one that I like a lot, first position U, second position U, third position C, UUC, that codes for phenylalanine. And AAA, so A in the first one, A in the second one, A in the third one codes for a lysine, and so on. So basically, if we get the three letters with this table, we can find out which is amino acid that's going to be put over there. So the messenger RNA is a sequence of letters that comes in triplets, no, ta-ta-ta, 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 and each three letters code for one amino acid. By the way, typically, the first amino acid in a prokaryote protein is going to be composed by A, U, G, AUG, that is methionine. Methionine is always or almost always the first amino acid in a protein in prokaryotes. So it's also coding for start. The protein starts in there. Okay, AUG, AUG is going to be coding that. So this here is the genetic code, and what's interesting here is that the genetic code in this example, it's redundant. We have more possible combinations, 64, that are necessary to code for 20 amino acids, so we have leftover codes, no more codes than we have amino acids to code for. We need 20 amino acids and stop. We have some stops here, so we have 21 requirements, and we make 64. So the genetic code is redundant, which means that the genetic code is degenerate. The word used for this redundancy is that the genetic code degenerates. For example, UU, A and UUG code for leucine. CU anything code for leucine as well. See, there are 6 possible combinations of letters that are going to be coding for leucine. The genetic code is redundant. In some cases, you have more combinations of letters, more codons, than we have amino acids. Of course, there are exceptions. For example, methionine, there's only one, is the AUG. There are others that you have only one. If I'm not wrong, tryptophan over here. No, okay. So that some have only one, but some are going to have many. Some have 3, 4, 5, 6. Now, leucine have 6 possible combinations. So the genetic code redundant degenerates. Okay. Just to illustrate to you, this is an exercise that we're going to be doing in class. We have here a piece of DNA. Okay, it's one of the size of the DNA. So one of the things that I might ask you is for you to go there, think this is a non-template DNA, and I say, why don't you make the template? So the other size of this DNA. And when you have the template, then I will ask you, why don't you make the RNA, the messenger RNA? So then you get the complementary and make the messenger RNA. And once you have the messenger RNA, then I'm going to say, okay, why don't you make the protein with that? So we are going to be talking about proteins and making all the stuff now that we can make. I'm not going to be giving spoilers. We are going to see that in class, okay? Opa. Later on, of course, we're going to be talking about mutations when you make mistakes, but also not spoiler right now. What I want you to take a look at now is in this piece of machinery here. This is a ribosome. Ribosome is composed by 2 subunits. You have the small subunit and the large subunit, and they both together, they make the ribosome that's going to be making the translation. Basically, this is the guy that reads the messenger RNA and attach amino acids together, making the protein. The ribosome, by the way, is a weird molecule because it's made by proteins, which here is shown in purple, but also is made by rRNA. The ones in pink and yellow, this is RNA. This is a weird molecule that's composed by protein, which is typical for making shape stuff, but it's also made by RNA, which is also possible to make shape with that, and it's quite good to be fitting RNA inside to be reading and doing all the stuff that the ribosome is going to be making. So ribosome, a molecule composed by proteins and ribosomic RNA. Okay? If you take a look on a ribosome, it's going to be having these 4 sites. One of them is the messenger RNA binding site. Basically, is a place where the messenger RNA slides through to be read like in a scanner. And the ribosome also have 3 sites. The amino acid site, or A site, or acceptor site, is the place where the amino acid goes. You have the peptidol or pepsil site, also the protein site, where the protein is built, where the amino acids are glued and they stay over there. And we also have the E site, which is the exit site, where whatever is bringing amino acids, it exits through the exit site. So we have this EPA, EPA, exit, peptidol or pepsil, and the amino acid or amino acid site. Ribosomes, 4 spaces inside of it. Here you have another view. We have the messenger RNA that's passing here through the messenger RNA binding site. We have here the EPA. You have the exit, the peptidyl, and you have the amino acid. So exit, peptidyl, amino acid, and you are building here a protein. Usually it's built in the interface between the amino acid and the peptidyl over there. Here we have a diagram showing a ribosome reading a messenger RNA, and on the left of this ribosome, a little protein is being formed. 3, 2, 1. Oh, there it is. Let's take a look again. This is quite nice. So let's see again. 3, 2, 1. And you see, the ribosome goes reading the messenger RNA and goes making proteins that is living here through the left of the ribosome. This is the process that's built. Okay. The molecule that makes it possible for bringing amino acid is also an RNA. And this one is called a tRNA, or transport RNA, or transfer RNA. It's an RNA which function is to transport amino acids. So this, we have here 3 different views of the same molecule of tRNA. The classic view that you're going to be seeing many exams and textbooks and stuff is this one that looks like a weird cauliflower. And it contains, on the top here, the amino acid attachment site. Basically, is this GCC over here, is a 3' and it connects an amino acid over there. So this is the guy that's going to be carrying an amino acid over there. You have these places here that helps it to fit into the A site and the P site. And in the bottom, we have a very important piece, which is the anticodon. This is 3 letters which are complementary to the messenger RNA, and it has the opposite of the code that is read that we saw previously in that table. The anticodon allows the tRNA to fit exactly in the complementary 3 letters of the codon in the messenger RNA. Here we have another view where you have here the amino acid goes glued over there. This is a more or less 3-dimensional view. And we also have here the anticodon. And a simplistic view where you have the amino acid gluing over here, and you have the molecule over there, and you have 3 letters over here in the bottom, which is the anticodon. Those are 3 different views of the transfer or the transport RNA. Okay, so that is the guy. And now let's take a look on how it works. So here we have a tape of messenger RNA. It starts with an AUG, AUG, and goes on and on and on. And we have there transfer RNAs that I'm representing here in this weird J's in a very simplified form. And the idea is that one of those guys are going to be able to fit in here. Basically, one of them is capable to fit with its complementary anticodon into this codon here, AUG. So if you need, pause the video for a second and think which of those are going to be fitting in there. AUG. Okay, so now that you took a look, you're going to see that the one that fits is this guy over here. A pairs up with U, U pairs up with A, and G pairs up with C. The complementary goes over there, and the first amino acid brought was MET. As I told you, MET is the first one in prokaryotes, so this is the first one that's read over there. After that, we have an UUA. What pairs up with UUA? So take a look around and say, yeah, okay, what pairs up with UUA? AAU, UA, UA, AU. they pair up over there, and then inside the ribosome, a peptide bond is going to be made here, and now we have a G peptide. And then ACC, what bonds with ACC? Yes, now you're getting good on this. Probably Red said very quickly, oh yeah, UGG. And when that guy enters inside the ribosome, another peptide bond, now you have a tripeptide. And then comes the GAG. What's going to be bonding there? Yeah, it's the last one over here. CUC, it bonds over there, and you make a peptide bond over here. So we just made a little string of 4 amino acids glued together, making a little protein. This is the idea of making. But of course, after the amino acid is attached by the tRNA, the tRNA goes away like this. Okay, for every single one, it enters, attach, go away, enters, attach, go away in the exit point. So these guys on the bottom there are the codon, 3 letters. So we saw this already. It codes for 1 amino acid. So here we have, there they are paired, just like I showed you, codon, and in the top we have here the anticodon. Okay, those are the anticodons, which are complementary to the codon. Just to show you how it works, MET, all proteins in prokaryotes start with a methionine. So I'm going to be having there the P site, peptidol site, where the letter that enters in there, AUG, is going to be an UAC. So MET is the first one. Okay, it starts like that, and now we have the amino acid, or the A site, the acceptor site, free with UUA. UUA fits AAU, so this guy now is going to be coming here, and it enters over there. Bah, it entered. In the moment it enters, it does 2 things. It's going to be making here a peptide bond, and this guy over here moves one movement like that, goes to that place over there, and at this moment, the guy that went to the E site, this transfer RNA, goes away, and we have now an ACC free to go another one. ACC goes UGG, so let's put the UGG over there. The UGG is going to be entering, and peptide bonds, and moving the ribosome one more place over there. So there it is. So this guy is getting over there. The transfer RNA that was there in the P now is in the exit, goes away. Hey, there it is, it went away. And I have a GAG, that allows a CUC to enter. So CUC is going to be entering here, and I have peptide bonds, and this guy here is going to be moving one more position over there, and this will carry on and on and on with the transfer RNA that have already given the amino acid living over there, and this will go on and on and on until you get to a stop, or until you get to a terminator part of the messenger RNA. So this will go on and on and on, and we are making proteins over there. Okay, so this is the idea. This is just repetition. I'm not going to be showing this again. Okay, that's the idea of making this process. So here, just diagrams to illustrate to you. We start with a MET in the initiation. It's the start codon. Okay, so here we have the MET. Then it's an empty space for a guy to enter here. Now here is entering a purple. It will go over there. So we have a purple over here, and they are going to be making the covalent bonds. After that, the one that was the green, look back again. The green that was here, now it is in the exit, it goes away. The purple that was in the A now is in the P, and now we have a free space for another one to come here, go over there, make the next peptide bond, and this will go on and on and on until you get to a stop codon. When you get to a stop codon, this stuff stops, the protein is let go, the ribosomes, they dissociate, and the protein is done. This is the process of translation. Okay, so this is it. Here we have things happening around. Now lots of information, lots of a diagram here showing all this stuff. And what's interesting here is the following. This is a diagram of a polysome where we have 2 tapes of DNA that are open and being read, and then one of them is making messenger RNA over there, and the messenger RNA have little balls that are already making proteins. So this all, in prokaryotes, they all happen together. DNA is open, make RNA, the RNA is already read and make proteins just on the following, okay? And here we have an eukaryote, probably, polysome, where you have ribosome with we have a messenger RNA with ribosomes reading and making polypeptides. The ones that are here on the end, they have already little proteins over here getting out of the ribosomes, and the ones that are in the beginning here, they shouldn't make much. So basically, it's this process you have here. Here you have it long, here you have it small, and you can have many ribosomes reading one piece of RNA at the same time. One messenger RNA don't make one protein. One messenger RNA makes proteins and proteins and proteins and proteins, hundreds, thousands, millions of them, one after the other in ribosomes working frantically, okay? This video is quite interesting from McGraw-Hill. I strongly advise you watch it because it will show you all the steps in the process, you show transcription, you show translation, and everything will be rounded up. And then we can have a good, nice discussion when you come to our class, okay? I hope you have enjoyed. Take a look, do the quiz to see if you grasp the main concepts, the most important ideas, and we'll see each other in class where you can have a nice and fruitful discussion about this entire process of using genetic information to make proteins. Cheers, bye-bye.